Hi. So, we have learnt a lot about the antibody, and now you know that there are a variety of antibody. There are subclasses which we call it as a isotype, okay, and there are further in some cases there are further some types, okay. And if you see this molecule, it is a very interesting and elegant molecule. The reason is that this molecule has a variability as well as constant region is also there. So, all these things are very good for understanding, but if you look at what is the genetic basis for encoding such molecule, it is a massive challenge, okay. And this challenge was there in the field for quite long time, okay. And this if you see, there are few very important points are there. There are some dilemma before understanding this the genetic basis for these antibody production. It was quite difficult, and still there are so many things which are under a very active research. But the most important thing is that you know that our genome is basically encoding about approximately 30,000 genes which encode protein, okay. So, in this scenario, when the genome is encoding only approximately 30,000 genes, in this scenario, how the antibody, the diversity is generated, okay, which is in very big scale, in very big order of a scale, okay, it is million to 100 million, okay. By this such a limited number of genes, okay. So, this was a quite interesting and big challenge in the field of immunology, okay. And what causes the difference in amino acid sequence? So, here you can see that the immunoglobulin has a variable region and constant region, okay. And this is also again changing from one class to another class. So, the variable region is remain constant and there will be another constant region. For example, if the antibody is generated against some antigen X, so the variable region, the first variable region will be in light chain and heavy chain, and the constant part of these antibodies first will be IgM, or it could be in case of light chain it could be a kappa or lambda, right. But this IgM having the same variable region and change to the IgG. So, this is very interesting, this is very complicated to understand, okay. And how does how can different heavy chain constant region be associated with same variable region? That what I was explaining, okay. How this is genetically taking place, these are very big challenge in the field, okay. And if you consider the simple theory of genetic, one gene, one polypeptide, then this is not fitting well with this kind of observation, okay. This kind of observation means then there will be a huge number of gene and the cell cannot accommodate that many gene, right. So, this was a very interesting, okay. So, in germline DNA multiple, so to answer this thing, in germline DNA, okay, there will be a multiple gene segment which is coding portion of this immunoglobulin light chain and heavy chain, okay. And all these things are basically taking place during B cell maturation, and there are different stages of maturation, okay. And then there will be a antigen stimulation okay, and then these gene segments are shuffled among all these. There are gene segments, I will show you in a short while, okay, and they shuffle and leaving coding sequence, and only one functional heavy chain and light chain are towards end. It will be there, and that will encode for the protein. You will see how it is taking place. I am not going to take you in very fine molecular detail. It is a very complex, okay. It needs a lot of time to understand, okay, but I will stimulate you to learn by yourself how this thing is taking place. There are several good books, you can go through that, okay. So, when this is taking place, then one concept is basically standing wrong. So, we believe that the genome of individual is same, right, in all cells, but when this is happening in the host, then the genome is not same. This gene segments are present on DNA, and this there is a recombination of DNA is taking place, okay. So, therefore, I can very confidently say that the chromosomal DNA in mature B cells is not same as other germline DNA, okay. So, in general we say that the DNA in all cells are same, but this is an exception. In lymphocytes, in mature lymphocyte, the DNA is not same as other cells. If you take the liver cell and if you take the mature B cells or T cells, then the genome will be different, and this is the basis for the discovery of this diversity, okay, how the antibody diversity is generated, okay. And there are so many workers involved in this in finding out the genetic basis of antibody diversity, but among all I am just talking about the key work of Susumu Tonegawa. He basically found out that there are the gene segments, and these gene segments shuffle among the in the genome, and then that will make a mature DNA, and then this will transcribe and then translate, and then that makes a antibody, okay. So, his work, I have seen his original paper. It is a very complicated work. It is a extremely hard work of his group, okay, and by a lot of experiment. At that time, not so much sophisticated Southern blot or RNA isolation methods were there, but he has done all those experiments using radioactivity, and then finally he established how this genetic diversity for antibody is generated. And for this great contribution, he received the Nobel Prize in 1987, okay. So, I will not discuss about his paper. I will just give you some glimpse, a very short glimpse about his work, okay, only one slide, okay. So, if his theory, initially his hypothesis was that, then how he can prove it? It is very simple. Here he has isolated the DNA from say another cells. Another cells means non-lymphocytic cells, and he also isolated the DNA from lymphocytes, okay, the B cells, okay. And then he performed restriction enzyme digestion, and he probed with here you can see that he made a probe for one sequence in this which is identified from mature B cells, okay. And then he, after probing, he found out that the genome is not same, okay, and that leads to the discovery of he initiated the concept of gene segments, okay. So, So his work is very huge. It's a lot of work, but I'm not going to take all those things, because if I will discuss those work, then it will take a quite long time. Okay, so I'm straight away jumping on the antibody diversity from his work, and later on many immunologists followed his work, and then several things are discovered. Okay. So this was the basically experimental basis for diagnosis of rearrangement in an immunoglobulin locus. Okay. So here I am giving a very simple schematic for B cell development. As you know very well, the B cell development is taking place in bone marrow. Okay. Once it is mature in the bone marrow, and here there will be a clonal selection and clonal deletion. I have explained you earlier, clonal selection and clonal deletion. Once this the B cell is mature, okay, mature means they will be naive. They will be naive. They have not seen the antigen. It's an antigen independent process. So maturation of B cell in bone marrow is antigen independent process, and once they mature, they are called as a naive immunocompetent B cells. Okay, and they basically see the antigen first time in peripheral lymphoid organ. Okay, and then they further mature and they start secreting the antigen specific antibody, and later on there will be a class switching. Okay, class switching means there will be a, for example, the B cells is producing only IgM. So from IgM to IgG, IgA, IgE, they change the classes. Okay, so this we call it as a class switching. Here you can see that most of maturation take place in the bone marrow, and antigen dependent processes are taking place in peripheral lymphoid organs. Okay. So by the Susumu Tonegawa's work and other immunologist work, they found out that for light chain, okay, there are 4 gene segments. The 4 gene segments encode for light chain. Okay, that is L. L is nothing, it's a simple leader or peptide sequence which basically direct the polypeptide to a particular organelle. Here you can see that this is a endoplasmic reticulum. Okay, so if you see carefully, then so there are 3 gene segments which basically decide the sequence of light chain. Okay, and this sequence, this region we call it as a VJC. Okay, V stand for variable, J stand for joining, and C stand for constant region. So the whole light chain is encoded, initially encoded by 4 gene segment, that is LVJC. Okay, but after maturation of this polypeptide, this will be having only sequence amino acid sequence derived from V, J and C. Okay, and in case of heavy chain, there are one more gene segment is there. Okay, besides L, V, J and C, and there is a one more additional gene segment is there which we call it as a D. Okay, D stand for diversity. So L stand for leader, V stand for variable, D stand for diversity, J stand for joining, and C stand for constant. Okay, so there is a recombination taking place in both light chain and heavy chain. Now we will see how it is taking place. Okay, here I am just showing you in a very simpler way the genetic organization of immunoglobulin gene segments. Okay, in germline. Here you can see. Here one more piece of information I would like to give, that this, in case of human, the lambda light chain is present on chromosome number 22, and in case of mice it is 16. and kappa light chain, all these gene segments are present on chromosome number 2, and in case of mouse it is 6, and heavy chain gene segments are present on chromosome number 14, and in case of human, and in case of mice it is 12. Okay, this is just a information you may need. Okay, so here I am showing you the arrangement of these gene segments. Okay, here you can see that there is a V lambda. Okay, V lambda, here you can see there are various J sequences. Okay, and there is a various constant region sequences. Here you can see the presence of these gene segments over the DNA. Okay, in case of lambda chain. Okay, this is for kappa chain. Here you can again see that there are various V sequences and J sequences are there, and there is a constant region. Okay, in this slide. And in heavy chain, for heavy chain, again you can see there are various variable region gene segment, and diversity region gene segment, and constant region. Okay, so here you can see that the constant region is present, and here if you see little carefully, there is a the constant region is encoding IgM. That's probably the first antibody which is generated is IgM. Okay, this is present in sequence. Okay, and then there will be a delta which is present on B cells. Okay, in mature B cell, and then there will be a production of IgG, and this is subtype is IgG3. Okay, like that. So this is the arrangement of various gene segments. In case of light chain, there is a variable region, there is a joining region, and constant region. So it is shown in this schematic. Okay, so you can see that this heavy chain has a various V, D, and J segment and constant region. Okay, this is the arrangement of gene segment over the DNA. Okay, now I will talk about the variable region gene rearrangement, how this gene rearrangement is taking place. Okay, so variable region gene rearrangement occur during B cell maturation in bone marrow. Here this is, you have to note it that this is antigen independent. Okay, and heavy chain variable region gene rearrangement takes place first. Okay, thereafter the light chain variable region gene rearrangement is taking place, and in the end B cell containing single functional variable region is there in DNA sequences. Okay, and heavy chain rearrangement basically the class switching happen after the stimulation of B cells with antigen. Okay, here you can see how this gene rearrangement is taking place. If you remember the previous slide, the first rearrangement taking place on heavy chain, and you have seen there are variable region, there is a diversity region, and there is a joining region. Okay, or joining. So these gene segments are there. Okay, so these gene segments are basically present over the genome, and the first rearrangement is taking place between D segment and J segment. Okay, so here you can see that the first DJ joining is taking place. Okay, and after that there will be a V DJ recombination is taking place. Okay, V join with DJ, which is already rejoined. Okay, means previously it is joined. Okay, so finally first DJ recombination is taking place, and then VDJ recombination taking place. Okay, and eventually there will be a again joining of this, the rearranged VDJ will take place with the constant region gene. Here you can see that there is a primary transcript. Okay. So, initially there is a DJ recombination is taking place, here you can see in this slide. Okay. And then this VDJ joining is taking place, and then this rearranged VDJ is joining with a constant region. Okay. And then there will be a formation of mRNA and polypeptide and so on. So, okay. In mature B cell, both for mu chain and for delta chain is taking place. That is why on mature B cell there will be a expression of IgM and IgD is taking place. Okay. Thereafter, the light chain rearrangement is taking place, and this light chain rearrangement is basically the recombination is taking place between V and J, because there is no D gene segment which is encoding for the light chain. There is no D gene, okay, D gene segments, okay, and the process is almost similar as previous, and then the mature mRNA will be generated, which is having a sequence for the leader sequence, okay, and V region, J region and C region, okay. And finally, the polypeptide will synthesize. You know all those things, how the polypeptide is synthesis taking place. You have studied the molecular biology, okay. So, all these things eventually result to the generation of antibody diversity. So, but in addition there are so many things which is taking place in formation of antibody, okay. How this generation of antibody diversity is taking place, this the way I have explained you, this is only one way, but there are some more ways by which diversity, specificity and high affinity generation of antibody, all these things are taking place, okay. Yeah, the first one is multiple gene germ-line gene segments are there, that is of course giving the diversity as I have explained you in previous slide, okay. There will be a combinatorial VDJ joining by VDJ. So, there is a some very important enzymes are there which is triggering this recombination. Generally recombination is not a very common phenomena, it's a very rare phenomena, and the enzyme for this this recombination is present in these lymphocytes, okay. And the enzyme is VDJ recombinase. Basically it is consist of two major enzymes. The two major enzymes were discovered, that is recombination activating gene. In short form we also call it as a RAG1 and RAG2, okay. If you make the knockout of this RAG1 or RAG2, then these mice will be immunocompromised, okay. This is very important enzyme, okay, and this is needed for this gene rearrangement, okay. And in addition there is one more enzyme, this is known as terminal deoxynucleotidal transferase. So, all these enzymes are basically playing a very important role in recombination of these gene segments, okay. There is a junctional flexibility. So, I will tell you briefly about the junctional flexibility in subsequent slide. What is the junctional flexibility? Basically, if you know that this codons are triplet codon, and if there is some addition and removal of these codons, okay, then the whole frame will change, okay, or it may change the particular amino acid. So, all those things are also there when the antibody diversity is generated, okay. There is some specific phenomena is taking place, that is P region nucleotide addition, and this is basically taking place via some unique sequences, okay. There is a recombination signal sequences. This recombination signal sequences ensures that there will be no two V region or gene segments will be present in mature transcript or during DNA rearrangement there will be no 2J or there will be no wrong recombination is taking place. This is ensured by this RSS sequences. Okay, I am not going to tell the detail. If I had a long time, then I could explain all those phenomena at a molecular level. Okay, it's a very interesting. There is a N region nucleotide addition which is also taking place via this RSS sequence. Then there is a somatic hypermutation. If you remember the antibody structure which I have shown you in previous session, I have shown you some blue strips over the light chain and heavy chain. Okay, not blue, it's a black color strips, and I told you this is a hypervariable region or CDR, complementarity determining region. Okay, so over there is a some finer mutation is taking place in order to make a high affinity antibody, and this is basically triggered through this somatic hypermutation. Okay, there will be a combinatorial association of light chain and heavy chain, and overall this will give a huge diversity, whatever the question people had earlier. Okay, how this there will be a huge diversity with a limited genome. Okay, in addition, you know that so far I have discussed whatever I have discussed. You did not consider the ploidy of cell. Generally, our cells are diploid. Okay, so how it is ensured that only, okay, what is going on in if it is the diploid case? So far you might not thought about that there are two sets of gene. Okay, so basically it is taken care by this very interesting phenomena which we call it as allelic exclusion. Okay, so first the heavy, here you can see that there will be a these gene segments are coming from paternal side as well as maternal side. Okay, so first it is okay, I will show you this schematic. Here you can see that progenitor B cells. Okay, so there will be a DJ recombination taking place in one of the allele. Okay, and if it is successful, then it will move to the another, but if it is unsuccessful, then since the cell has another copy, so over there this DJ recombination is taking place. Okay, so in that way this whole thing is taking place, and if once this mature or proper transcript will be generated for light chain and heavy chain, then that will the another set will be not used until both set are exhausted. Okay, so this is a model to account for allelic exclusion. Okay, there is a junctional flexibility. I have told you in previous slide what is the junctional flexibility. Here you can see that there is a different possibility how this once this gene, the DNA will break, this can combine with various bases on another gene segment, and that may result, sometime it may result to the productive or sometime it may result to the non-productive. Here you can see in first three cases there is a productive, but another two cases it is non-productive, because that results to the incorporation of a stop codon and polypeptide cannot be fully synthesized. So this is a non-productive. So if this is the situation, if the cell will face this situation, then the cell will move to the another set of gene, another allele. Okay, so in that way this allelic exclusion work. Okay, now there is a one more question, last question, which is how the class switching is taking place. Okay, so there are some still it is under quite active investigation, but still people are trying to find out how the class switching is taking place, but we have some evidences which helps in understanding the class switching. Okay, there is some sequence present in upstream to the constant region gene segment, which we call it as a switch region, and it is consist of about 2 to 10 kilobase pair. there is a switch recombinase, there are enzymes, and in addition there are some factors like IL-4 which is produced by Th2 cells, they also help in class switching. Okay. And finally, there is a discovery of one enzyme which is playing very important role in class switching, that is activation induced cytidine deaminase, we call it as a AID. Okay. It is simply RNA editing enzyme which changes C2U. Okay. C2U and that if you make the knockout of this mice, then there is a no class switching in antibody. Okay. So these are the things which triggers the class switching. Class switching, you understand, from IgM to IgG type. Okay. Here overall how the antibody diversity is generated, you can understand. So there are this is in case of human. So there are in case of human there are 48 V sequences are there, okay, for heavy chain, and 41 V sequences are there for kappa, and 34 for lambda. D sequences is present only in heavy chain, so this is 23. It is not present on light chain, I have explained you, and J sequences are about 6 in case of heavy chain, and for light chain kappa chain will be 5 and lambda will be 6. So if you do simple mathematics, you can find out that and there is a we can make a huge number of antibody, okay, about 2.5 million types. And here please remember that you have not included the somatic hypermutation, J joining and all those things, okay, junctional flexibility and all those things. So if you add up all those things, so there is a the cell can generate a numerous diversity. It's difficult to count. Okay. So this is the genetic basis of antibody diversity. This is a very important topic. Okay. Or very important important discovery in the antibody, or in fact the immunology. Okay. And similar diversity is also taking place in case of T cells. Okay. Which I will discuss in next session, and I will take a very short session about the T cells, and then we will finish this week. Thank you.